Okay, so we left off in the middle of systematics. Middle of section 2. So, not yet. I will. So phylogenetics rely heavily on data about characters that are either present or absent in taxa. If you cannot see the characteristics or define them, or if it's a fossil, sometimes that's a problem. But other kinds of data are also important. We compare many kinds of evidence and apply logic carefully when we infer phylogenies, meaning when we determine what that species or family or class is. So scientists also revise phylogenies based on new evidence. So when I do a DNA fingerprint of something and I realize that it is related to another thing, I don't just ignore that. We add that to the current taxonomy and phylogeny. And then something called morphology. This means the shape or the changing structure, the study of. But it's looking at the physical structure or the basic anatomy of organisms. There's large-scale morphological evidence like seeds and flowers, and we talked about how we put those different types of plants into clades based on that. And scientists must also look carefully at similar traits to avoid using analogous characteristics for classification. So if I use the wings, for example, again, of an insect and the wings of a bird, though they are both wings, they do not have a common ancestry. It would be a mistake to infer relatedness with those two organisms. Fossils, that's a big one with. You can accidentally make a morphological mistake very easily looking at fossils. Now, an important part of morphology in multicellular species is the pattern of development from embryo to adult. And organisms that share ancestral genes often show similarities during the process of development or how they develop. We looked at vertebrates. We've seen how all vertebrates basically have the same basic embryological steps. So the jaw of an adult develops from the same part of an embryo in every vertebrate species. We all have one of these, a mandible, the lower jaw. I challenge you to name one vertebrae that does not have one. It's tough. Yeah. So scientists can now use genetic information to infer phylogeny. Hello. Oh, but we're going to see you, right?
We're going to see you.
We'll see you guys later. All right. Hey, your guest speaker was phenomenal. It started a little sketchy, but it was smooth sailing after. I'll tell you about it. Okay. Oh, we had a research material scientist came in and gave quite the talk today. What? Oh, yeah. Yes. So some mutations may be passed on to all species that have a common ancestor, but remember, it's the genes that are passed on from generation to generation, and you have to have mutations in those genes. And it can't just be in like the skin or the body cell, it must be in a sex cell of that species. Now, when I look at molecular evidence, the only downfall to DNA science is if the species is extinct, and I'm trying to look at our past. Like if I go back in time and I find a pile of trilobite fossils, I can't do a DNA fingerprint on them. So genetic sequence data are not used widely for cladistic analysis. And first, the sequence of DNA based on a gene or several amino acids in a protein is determined for the different species. And then each letter or amino acid at each position is going to be compared to the other. And so now I can compare the amino acid sequence of a human to a gorilla to a chimpanzee to a monkey and see the relatedness. Or a tomato to a berry, or a tomato is a berry, to a blueberry or to a strawberry. You guys know a tomato is a berry, right? Okay. So here we can, and this actually has a tomato in it, that's good. So here we can compare the amino acid differences, and anywhere you see yellow, that is a difference in amino acid sequence from one to the other. And you can show the actual evolution of an organism and basically sequence it by the numbers of differences in their amino acids or their DNA structure. So we can do this with species of people. You can even do it with subspecies and seeing how long ago they diverged and became separate races. In our case, we call our subspecies. Now, at the level of genomes, alleles may be lost or added over time, and another form of molecular evidence is the presence or absence of specific alleles or the proteins that result from them. So from this evidence, the relative timing between genetic changes can then be inferred. So we cannot predict where the mutations will occur, but we can predict when they will occur and at the rate at which they will occur. Even if you're exempt from the exam, please do take the notes. I might be tempted to give you a test if I don't see everybody actively engaged. So cladistics can determine only the relative order or divergence or branching in a phylogenetic tree. What's up? Okay, that's okay. That's okay. I wasn't talking to you. So using cladistics, scientists have identified lancelets as the closest relative of vertebrates. They're the closest thing we can find in the fossil evidence of something that almost has a vertebral column, but not quite. They remind me of kind of a lamprey creature. Now, the oldest known fossil of vertebrates are about 450 million years old, and the oldest lancelet fossils are 535 million years old. That means that I can't tell you the exact age that vertebrates evolved, but I can give you kind of a range. We know it's between 450 million and 535 million years ago. We know that they must have actually diverged before that point, and so we know that the divergence had to occur before that 535 million years ago. So DNA mutations occur at relatively constant rates, so they can be used as an approximate genetic clock. Again, we do not know what mutations will occur, but we know the speed at which they occur at. And so scientists can measure the genetic differences between taxa and estimate time of divergence, so how long ago they diverged. We can do this with our own human ancestry. We can say, okay, this DNA has way more differences, let's say, in it. As if I look at Swedish DNA, I'd have to have somebody that's 100% Swede, but they do exist in Sweden. So I go there and I get their DNA. And then I get like a Native American who's 100% Cherokee, and I look at their DNA. And then I get a South African, and I get their DNA. And then I get, I'm probably missing a couple here, but you get the idea. So I take a bunch of these different samples of DNA, and I compare them to each other. Turns out that the one that is the origin of man is in Africa. Okay, there's actually one tribe there that is the original strand of the human DNA, the earliest human man DNA that we have. Now, it's changed a lot, but we can tell at the point of divergence when the other DNA became different from separation of land mass and geographic isolation. Yes. I don't know that off the top of my head. It's in African, obviously. I'll look it up for you. I went to a Howard Hughes Medical Institute holiday lecture on it one time. It blew my mind, really. I was like, oh my gosh. So you can trace them back to one region. But I mean, it's something we all should really know, right? Like, where did all our races of people come from? Now, they don't have the original DNA, right? But that is the origin. That's the oldest one we can find on the planet, and that's where it's at. Okay. I'll show you the phylogenetic tree here in a little bit. Okay. So modern systematics uses what's called the principle of parsimony. I call this common sense to construct a phylogenetic tree. Parsimony, you guys, just means take the path of least resistance, the one that makes the most sense. If you have, let's say, two differences in DNA between one plant and another and four differences in another plant, we say that the one with two differences is closely related to the original. And so that's called the principle of parsimony. So this holds that the simplest explanation for something is the most reasonable unless strong evidence exists against that explanation. So this does include using inference and looking at morphological structures as well. So given the two possible cladograms, the one that implies the fewest character changes between points is preferred. So again, common sense, right? If you have two differences and another one has 4 differences. The one with 2 differences is more related than the one with 4. Quick summary, I'll let you guys work on that big review. Scientists traditionally have used similarities in appearance and structure to group organisms. However, this approach has been problematic. Grouping organisms by similarity is often assumed to reflect phylogeny, but inferring that phylogeny is complex in practice. It's not always that easy. You can make mistakes when doing inferences. In cladistics, using those clades or those outgroups is used to select the most likely phylogeny among a given set of organisms, and we compare many kinds of evidence, including genetics, and apply logic carefully in order to infer phylogenies, always using the principle of parsimony. Merry Christmas, right? Biology is complete.